Hi, good morning, everybody. It is, as we always do, a weather report when we start. It seems like that's how I started every talk. But today is a sunny day. The clouds are gone. It's actually it's going to hit 70s, and it's a beautiful day outside. So the spring is here. The red buds have been blooming. The dogwoods are out. So if you get this weekend to go out, go hike somewhere in this our beautiful area, this would be the perfect time to do that. So those of you who don't know me, I hope by next year I'm not going to have to say this over and over again, but I'm Associate Vice Chancellor at University of Arkansas and the founding executive director for the Institute for Integrative and Innovative Research, I-R, as we say. But I think for today's purpose, I'm also going to say I am Distinguished Professor of Biomedical Engineering because we have a very somebody who's dear to my heart who's visiting today from Biomedical Engineering. So those of you who are on the web and perhaps on Zoom for the first time and you haven't heard it still, but I-R, we are committed to a mission of bringing convergence research together. And what we mean by convergence research is bringing academia, industry, the community, all of us pulled together to address what we call wicked problems, problems that are not easy to have no simple answer, are difficult to come up with a solution for, require you to be flexible, require you to take risk. And as I said in yesterday's talk that I was giving at another place, we deliver. We are very much interested in making sure that we translate things out and not just have them ruling in our labs forever and ever, but actually getting those deployed. And if we are going to get them deployed, we need our partners in policy, in economics, in political science, in behavioral health, all of those along with us in our ability to deliver. So we want to be the leaders in this. We're leaders in it. So we're working towards it. That building is coming along. So 5 words describe us: integrative, convergence, economic development, at scale, and societal impact. So keep remembering those. So today we are going to have I'm not going to tell the details of it, but we are going to hear about what happens when we do implants inside the body, and it's a hostile environment. And what happens to that tissue device interface. There is an expert with us here today talking to us about that kind of a technology end, but also clearly in order to be able to get things deployed, you have to get in the medical implant world, you have to solve these problems. Otherwise, things are not going to get deployed. They're going to stay within our labs, which is, and so we need to know what's the problem, but also what might be the solution. And so I'm very pleased to have with us today Professor Kevin Otto. He comes to us from the Department of Biomedical Engineering at the University of Florida in Gainesville, North. We know Kevin, I was in the South, you were in the North. And Kevin completed his undergraduate degree in chemical engineering at Colorado State. And then he got his master's and Ph.D. in biomedical engineering from Arizona State University. As you all know, I was there, but he was leaving with his professor. He was a grad student, and some of you grad students can relate to that. When your professor moves, you move with them, and then you have some courses left over and all of that stuff. So unfortunately, he wasn't there when we were there, when we arrived there. But that means that over the past 2 decades, we've known, because we knew his professor too, we've known of his work and his effort in moving the field forward. And he is very well recognized. He's, he's a fellow of AIMBE, is moving things forward. That is the highest possible recognition you could get, 2%, I think, or something, are recognized for that. And he's brought technologies forward, working in the DARPA mode, which we all talk about a lot. And so he knows what that means, and he's going to talk to us about it. He has a passion for exploring ways to improve the quality of life for individuals with neurological disabilities. Another one of an overlap for us, but I think the passion that we have of bringing about well health. So with all of that, and without further delay, I would like to invite Professor Otto. It's all yours.
Thank you. Okay, I'm all set up. Thank you so much, Renu, It's really an honor to be here. As Renu mentioned, I've known Renu and Jimmy for over 20 years, and we've intersected in many different dimensions many times. It's kind of funny, like how many different places we've been near or adjacent to, and working together, and especially in research. So that's what we're going to talk about today. I'm going to talk about this term optimizing, which for those engineers in the room, optimization comes with a very specific definition. And I already off the bat want to deflect criticism to say this is not my word. This is our NIH program optimizing devices for stimulation and recording, their word. So first rule of getting sponsored research is use the sponsor's language. So I'm trying to optimize in the NIH sense of the term, and we'll talk about that. Okay, first of all, though, I really appreciate the welcome here. I want to welcome you right back. We, together with Renu is the conference chair. I am a program co-chair for the IEEE EMBS meeting in the EMBC in July in Orlando. So convince your faculty, etc. Send you, please come. There is one more round of submissions available for abstracts for posters. So you can still get in, you can still present. I'm going to tell you 3 different stories today, 3 different little stories. And one really has to do with the brain and micro devices. The brain implants have been around for a long time, clinically being implanted in patients since the 90s and potentially before that as well in an experimental sense, but not necessarily at the micro scale. At the micro scale, there still aren't clinical implants. There's still exploratory work. And I'm going to talk about one of the reasons those haven't quite yet hit the clinical sphere is because of this device tissue interface. So I'll talk about our efforts to characterize it, to study what's going on. And I'll talk about one possibility to help ameliorate some of the challenges in that solution, in that space, with a term we call rejuvenation. We'll talk about usage of those devices for over 20. My dissertation work was on microstimulation of the brain. So try to drive pockets of activity to convey information. So we'll go through a microstimulation and the optimization of that, engineering the future. We'll then code switch back to spine and nerve, work that we've done in the periphery and in the spinal cord to develop devices, develop interfaces, using some of the information we've learned from the brain and studying those. And then finally, I'll take a brief foray into our work in bioelectronic medicine. I'll introduce that topic, explain what it is, a little bit of foreshadowing why I'm so passionate about it. It hits home personally. So that's a really fun way to sort of cap it off. Okay, so diving into the brain micro devices. Micro electrodes are good brain interfaces. For any of you out there that are familiar with the NSF scoring scale, you know that good is really not very good. Poor, fair, good, very good, excellent. So good, I'm using the term good on purpose. They are good. They have a lot of reason to look at them, to study them, to use them, and they have clinical applicability. They're going into human patients now all the time. But they have challenges. They have clear opportunities for improvement. We've seen these used in a prosthetic application. Here's a video of an individual who has electrode arrays into the motor cortex, as well as into the somatosensory cortex. The motor cortical activity is being used to drive the robot to move. He's imagining moving his arm. And the somatosensory cortical electrodes are being used to stimulate, to give him an artificial feeling of touch. So this is an individual named Nathan. He's driving that robot. Fist bumping President Obama. And when President Obama hits his robotic hand, the force in that, the pressure sensors there are translated into electrical stimulation of the somatosensory cortex. So he can not only use output, he can use input, microelectrode devices. This is a research grade clinical opportunity, largely driven by a company named BlackRock Technologies. This is also receiving a lot of press lately by this particular company, Neuralink, who happens to be started by someone sort of famous. But one of the reasons that I put this slide in particular is to indicate that the field is not as small as a lot of people are led to believe, or a lot of people currently think. So I've mentioned two, BlackRock and Neuralink. Medtronic and Verily, very big companies, have been in this space for a long time. Neurotech devices, neurotech different implants and interfaces for interfacing with the nervous system. So big companies like that, Setpoint Medical and Galvani, are two examples of bioelectronic medicine companies that are currently in clinical trials and development. You have hardware companies like Intan, software companies, OpenBCI, new startups that are coming out all the time, Forest, Neuronoff, Motif. Just a brief spattering of some of the kind of categories, but really when we look across the space, it's really rich. These are all the companies that are sort of out there right now that are in neurotech, doing something neurotech related. And this list is obviously not comprehensive. I'm happy to share this slide. I can give you another pointer to a resource that we've, actually one of my colleagues at UF has generated to help, especially students, with understanding the commercial landscape in this space. Okay, so if in fact you were to have a disease or an accident and you needed a microelectrode, why could you not get one clinically today? And one of the reasons I'll argue is because they do exhibit this reactive tissue response. Just like a splinter in your finger, after the splinter goes in there, if it stays in there, it generates responses from the body. Those responses are due to many different things: the size of the device, the mechanics of the device, how clean it is. Hopefully we're putting in clean things, but it turns out there's degrees of cleanness that many of us have not ever thought about. Or maybe how flexible is it mechanically? What material is it made out of? So within this space, we typically think of the reactive tissue response sort of like this, at least in the brain. If in fact we have a wired microelectrode, again, that changes the mechanics in this case because you're actually tethering, you're holding that in there. We could see just a generation, a cartoon of skin and bone and dura with the electrode implanted into the brain. And the little blue dots are sort of supposed to indicate neurons. Right after you implant, glial cells tend to come in and encapsulate the device. These are the first responders. Microglia, astrocytes are going to follow. There's cytokine storms locally that are coming in. It's actually sort of a toxic environment in the beginning, a very inflammatory, hyper-inflammatory case. That can lead to neuronal cell death and migration away from the electrode itself. One of the main factors in device performance is how close is that neuron to our electrode. So this, all of a sudden, is making our device less efficacious by the neurons either dying that we're trying to talk to or moving further away. It also results in an impedance increase. So just to define that term, impedance is complex resistance and reactance. If in fact we want to interface with those neurons that are out there, the glial encapsulation is creating an electrical insulation barrier that limits our ability to talk to those neurons. That's impedance, okay? And so as that layer gets thicker and tighter, the impedance goes up. And overall then an efficacy decrease. So how do we study this in our laboratory? We've, for about the last 20 years, we've used a device commonly known as the Michigan electrode array. It's marketed by a company called NeuroNexus Technologies. These are silicon substrates. So this backbone is silicon, made with the same techniques, photolithography, that they make computer chips with. The little metal sites right here are what we're using to interface. So in this shank alone, I'm showing you 4 sites on a single shank that could go down and give us 4 individual spots to listen or talk to pockets of neurons. There's 3 different packaging components, how you might actually use them to interface with them to get them into the spot that you want. We've done a lot of electrochemistry in my laboratory trying to understand how does the information that we're sending down these devices end up communicating with the saltwater environment of the brain. So for those of you that have taken second semester physics, you may remember, or first semester chemistry, electricity in wires is carried by electrons. And in this case, since it's actually silicon, it's carried, it's even more complicated than that. But you can think it's just electrons running down there. But electrons don't exist in free solution. So once the information hits the edge of that electrode, it has to transduce into a different form of electrical energy. In that case, it's an ionic current. Ionic current moving throughout the saltwater space that is the brain. Just like exchanging money at the airport, changing energy forms costs energy. It costs, there's a loss involved in that. And so the ability of a material to transduce that from an electronic to an ionic current has a big impact on how these devices perform. So we've studied different materials that can be used to change that electrochemistry, iridium oxide, the conductive polymer polyethylene dioxythiophene, which is called PDOT, or we put these in a sterile environment, as sterile as we can, so that we're minimizing the amount of foreign contaminant. Here's a picture of the electrode going into the brain, in this case, of a happy and healthy rat. We use rats primarily for a lot of our preclinical experiments in our laboratory. Okay, so I talked about the reactive tissue response. What does that look like? Here's a picture of a device implanted into a brain. This is the surface of the brain. This is the width of a Michigan microelectrode, about 100 microns wide. For reference, that's the width of a person with thick hair. That's the width of one piece of hair, if you have thick hair. Not my hair, I have thin hair. But if you have thicker hair, that's about how wide a thick piece of hair is. So it's stuck in there, and you can imagine the little sites are down here. We, in this case, we stained this tissue for different cell type markers so that we could study what cells is this device encapsulated by glial tissue? Is there a spatial distribution to that encapsulation? This device had only been implanted for 1 week. So the first thing we looked at was a marker for microglia. Microglia are the macrophages in the brain. They're first responders. They're not the neurons we want to talk to, but they do come in and try to attack the device. If the device was bacteria or cell fragments, they would eat it and then go get rid of it. And so you can see right here, they're trying to eat our device. This is called phagocytosis. In biomaterials literature, this is what's known as frustrated phagocytosis. They can't eat it. So when they go through frustrated phagocytosis, they eventually give up, link arms, if you will. They join membranes together and encapsulate the device in a total thin encapsulation of cell membrane. So you end up getting multinucleated giant cells surrounding the device. Membranes are very good at separating ions in solution. That's one of the fundamental things you're taught in the very beginning, is how membranes handle ionic transfer, keep the inside of the cell different from the outside of the cell. So this is a clear barrier to our ability to communicate from the device out to the tissue. It's not very set up yet. This had only been implanted for a week. You might also note it's a lot more encapsulated at the surface. That's going to be a theme. We're going to keep seeing that not only in anatomy, but also in physiology. The reactive tissue response and the degradation in performance is worse towards the surface of the brain. Okay, so we also looked at astrocytes. Astrocytes come in after the microglial response. They help to rebuild. They also help form the blood-brain barrier long-term. So you can see a nice blood-brain barrier here. This is a blood vessel. You can see a little blood-brain barrier here. They form tight junctions to help form part of the blood-brain barrier. And they haven't done it here yet, but you can see a lot of them are starting to turn. They start to turn and grow towards the device. And if this was 3, 4, or 6 weeks later, you'd see much more of this kind of response set up all along that device. The last thing that we did in this particular section of tissue was to look at oligodendrocytes. Oligodendrocytes wrap around the axons of neurons to help with communication. They're sort of insulation surrounding wire, if you will. And demyelinating disease is a traumatic aspect of there are many different diseases that contribute to demyelination. We know that inflammation contributes to demyelination. You can actually see pockets of demyelination surrounding the device. We haven't done a whole bunch more work on this. Other people have from here, studying a lot of how the glial response sets up in response to implants. Okay, so that's a very anatomical look. What does it look like physiologically? We've observed that can result in a limited lifetime. And so we published a paper here last year looking at the stability of electrophysiology recordings as a function of time. So this is a sort of quote unquote nice recording. And if it looked like that forever, we'd be happy. If it looked like that forever, it'd be flat. And so we can sort of quantify what is the amplitude of this waveform as a function of time. But remember that we have depth in cortex. So we can also look at it as a function of depth to see, is there a difference if we're up there in the reactive tissue response towards the upper edge or lower? And in fact, that's what you see. The amplitude is higher in the lower layers, layer 5 especially, and it tends to fall off as we go down. And in fact, if we look longitudinally, that's also the case, that the signal to noise ratio tends to go down, and it goes down faster for the surface areas, layer 1 especially. This is usually the point that a lot of people say, so what are you doing? What are you going to do to fix this? And a lot of people have put a lot of effort into fixing this, including DARPA running a $65 million program to address it. I'll tell you that we've written a lot of our perspective in review papers, starting with this one in 2014. And then within this Handbook of Neuroengineering, we published another couple of chapters. So I'm not going to go into a whole bunch of mitigation strategies here, but I will tell you about one in particular that we're excited about. And that is the idea of rejuvenating the electrode. Most of the approach so far has been to change the mechanics of the device, to make it smaller, to make it softer, to change how you insert it faster, or maybe change chemistry, try to reduce the inflammation surrounding the device, release some sort of drug. Those have all been really challenging to do, especially long-term. This potential technique gives us the ability to do it repeatedly locally, and it doesn't rely on any sort of outside change. The theory behind this, the basic premise, the theoretical framework, if you will, is that if we have the glial cell encapsulation and some level of neuronal cell death, if we're lucky, which actually ends up being a majority of the time, you don't have complete cell death surrounding the device, complete neuronal cell loss. You have neuronal cell loss, we have a slight decrease in density, but there's still neurons there. They might just be behind that glial cell wall. They're close enough to listen to, but we can't see them because the window's dirty. So what if we could just get rid of the glial tissue? Electrolytic lesion. This idea has been employed in many different situations. My favorite example is submarines use this to clean mussels off of the windshield. They electrolytically charge the windshield. Mussels use proteins to stick to the windshield of the submarine. And if you electrify the windshield, What it does is it breaks all those proteins that are adhered to the surface, and the muscles fall off as they're cruising along. So the idea has been out there for a long time. The question is, can we do it here? Can we do it safely? Can we do it repeatedly? Does it damage our device? Does it cause long-term damage to the body? So I'm not going to dwell on this too much, but for the electrical engineers in the crowd, people that are curious, I'm happy to talk about this at length later. One of the ways that we look at our effectiveness is through impedance spectroscopy. This is a Nyquist plot showing data from an impedance spectrum. You don't have to worry about axes or anything. Let me just tell you that this, especially the curved aspect, means there's a big reactive tissue response. The more curved, the more it's pushed up and to the right, the more encapsulation there is. We know this a little bit from Justin Williams in his dissertation. He measured this as a function of time. This is the same electrode measured over the course of days, and you can see that develop over time. As the reactive tissue builds up, the curves get pushed out to the right, and they get more and more curved. We can model this so that we can try to put different aspects of this data into different compartments. Is it the electrode changing? Is it that immediate protein layer changing? Are we blasting the proteins that are adhering to the electrode? Or is it something around the cells or through the cells that's changing? Can we understand this? If we see changes, can we put it in these compartments? So what we did is we applied the energy to the electrode. We used voltage pulses. They're a little high right now, and that's part of the optimization. We have to figure out how to decrease that or at least optimize the energy. But you can see that we essentially, quote unquote, reversed aging. We rejuvenated the electrodes. We took this and pushed it back to where it was when it was implanted. Okay, so what, right? This is not going to drive your wheelchair or when you get to meet President Obama. It's not going to move your arm. But this will. This is right before, and this is right after. Those big spikes are neuron action potentials that we're recording on electrodes that were dirty. We cleaned off and then recorded action potentials on those electrodes right there. So we rejuvenated. And this is sort of a snapshot, right? But it does, there are many aspects that do work. You can do it repeatedly over time. This is the signal to noise ratio before and after every day in a single animal over the course of 150 days. So come in, plug in the animal, recordings are dismal. I'm a postdoc, I'm never going to get a job. It's horrible. Rejuvenate, and up here you can actually do some science. You can go forward with that. So you will note this line is not flat. We're not solving the problem, not yet. It's going to take a little more optimization in this space. But this technique, that's the effect that it has over time. We started with our optimization looking both at changing the waveform energy. Is there ways that we put it in that it's safer, that we might make the line flatter? And we're looking more at macro electrodes as well to see if there's more of a clinical indication for things like a deep brain stimulating electrode or a surface electrode. We looked first at a segmented DBS lead and secondarily at a surface ECOG lead that would sit on the surface of the dura or immediately below the dura. And in this case, we see the same thing. It starts out here, after rejuvenation goes down to here. Over the course of 7 more days, it goes back up. We weren't rejuvenating every day. So you can see that as the electrode becomes more and more encapsulated over time, it comes back. And we looked at different frequencies. Again, going back to that, trying to optimize. How can we figure out, before we were using DC, which is generally not a very great idea. It works, but we need to make it safer. AC will be safer, but we need to figure out what parameters in the AC space work. And so this is data for the segmented DBS lead. This is data for the microECOG lead. And you can see we're starting to understand what frequencies seem to have the biggest effect. More work to do here. We'll keep asking study sections to let us do that work. We're still not successful yet, but we will be. Okay, so we'll switch. Let's talk about microstimulation. Can we use these devices to stimulate the brain to, you saw the person receiving the stimulation and feeling that stimulation, feeling that touch. It is a powerful information transfer mechanism. Over the past 20 years, this was my dissertation work. We've done a lot of different work studying single electrode microwires in rats, in auditory cortex, published a lot of work in those spaces. When I started my first faculty position, we started to look at different materials. Here's the polyethylene dioxythiophene, PDOT as a material. We looked at longitudinal aspects. What about over time? How does it work? We looked at changing waveform energies, asymmetric versus symmetric waveforms. We also did a lot of work collaboratively, some with Sleeman Benzmea, doing that in primate models over the course of time, as well as looking at surface electrodes with Justin Williams's lab at Wisconsin. But let me give you just a little snapshot from our group lately. Microstimulation is susceptible to the same degradation, and we think it's likely due to the same mechanism. How do we measure that in my group? This is our basic technique. We use behaving rats and ask them, did you feel that? Did you sense that? Did you hear that? We're asking them questions in a behavioral space, and it's sort of like going to the optometrist, for those of you that are wearing glasses out there. 1 or 2, 1 or 2, 1 or 2. That technique is very similar to the technique we're using here. It's also sort of like the old GRE test. We start out with really easy questions. Every time they get it right, we make it harder and harder and harder until they start to get at their threshold, and then they start to miss. This animal is water deprived when they come into the box. They haven't had water for 20 hours or so. They're thirsty, they're ready to drink. They come in and they see this spout. It looks just like their spout at home. So they're ready to drink. But we have the spout instrumented so we can tell when they lick. When they lick the spout, it shows up on the oscilloscope like that. So that's a lick. When we first train them, we train them with the acoustic sound. And so they're licking, they're licking, they're licking. They hear a sound. And they don't know the first time, but that's a warning. Stop licking. There's going to be a shock. So the first time they get a little shock on their tongue. It's not damaging. It's not painful. It is aversive. They don't like it. And so I can say that it's not damaging. It's not painful. I haven't done it, but my students have. And they say it feels like putting a 9-volt battery on your tongue. For those of you that have done it, it tingles. It's aversive. It doesn't feel normal and natural. So you don't like it. You know it's coming. It's a signal. But it doesn't, your tongue isn't broken forever. And it's not painful. So they learn quickly when they hear the sound, okay, stop licking. These rats are very good at this. They don't waste any water. You'll watch, they just slightly change their lick pattern to game the system so that they're just, rather than lick, 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 they go, just go grab the water before it comes out of the spout. Doesn't matter. That's actually, I don't care how they do it, as long as we know that they heard the sound in this case. After they're good at the task, we implant them into a sensory cortice. Lately, we've been doing a lot of somatosensory cortex work to try to inform the human somatosensory cortex work that's going on. And then we replace that acoustic stimulation with electrical stimulation applied to the device. So that's going to show up on this yellow stream here. Here's the animal. It's going to start licking. Lick, 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 lick. There's the lick pattern coming in. Your job is either to watch the rats or watch the data, whichever you're more comfortable with. They tell the same story. I'll tell you when a stimulation happens. It'll come in yellow. And then there's the stimulation. It's on right now. So you may have noticed the rat pulled back a little bit, or you can look at the data. The rat obviously sensed that. And we know, okay, the rat sensed that amount of stimulation. The next time we'll make it lower. And then we'll keep going down until they start to miss it, and we can hone in on threshold. So that's our very typical threshold measurement. We can get somewhere between 5 to 10 thresholds a day. That allows us, remember, we have somewhere between 16 electrodes in an animal. And so if we're doing single electrode work, we can measure that over the course of time. Here's a paper that came out a couple of years ago that talks about long-term performance of this task. In this case, surprisingly, or not, it's layer dependent. And it's the same story. In layer 1, thresholds are going up, almost doubling over the course of 30 to 40 weeks post-implant, okay? Thresholds going up, that means we have to keep turning it up to get the same response. The animals are becoming less and less sensitive. Layer 2, the thresholds are going up, but they're not going up nearly as steeply. Layer 4, up, but again, a little bit less steeply. And then layer 5, they're almost flat, which corresponds actually incredibly well to the recording physiology that we saw. And we've done a little bit of work on characterizing glial cells. The layer 5 and layer 6 glial cells were the lowest. And in particular, the IBA1, which are the microglial aspects, are the lowest down here in layers 5 and 6. We've also found out that the cortical depth is key for physiology. My student in this case, a really, really remarkable PhD student, Morgan Urdaneta, he came up with this idea of tachyxy. So for a few of you who've been around for a while, you may have heard of chronaxy. Chronaxy has been around in the field of neurophysiology and electrical stimulation for 60 years. It's a while. But basically, what we wanted to know is, if we know the threshold as a function of time, are there other aspects of stimulation that are more sensitive at those layers? Does that sensitivity in layer 5 and 6 translate to other things? So in this case in particular, what he's looking at is stimulation rate. Is the animal more sensitive to rate down in those deeper layers? So what we found in this case, I apologize, this graph's a little small, but we found this is basically the sensitivity with depth, the same graph that you just saw. They're typically more sensitive down in layer 4 and 5. Their thresholds are lower. And if we look at the speed of the stimulation train, if we're stimulating very fast, meaning the current is additive, the action potentials are additive, we're actually putting more current in at that level. Thresholds go down with fast frequency. And at some point, you're at 2 times their minimum. And my student he called that tachyxy. Your tachometer on your car is how fast the motor is spinning. Tachyxy is, tachos is Greek for rates. So what we found is that the animals are actually most sensitive to stimulation in those same layers, layers 5 and 6. When we have the healthiest interface down there in the deepest layers, we have a lot more ability to stimulate with low intensities. And we have, if you use frequency as a programming method, meaning can we use different frequencies to put information in? Rather than using amplitude modulation, could we use frequency modulation to give energy or give information to the animal? We have the best ability to do that down there. Okay. So as a PI, I like to challenge the students all the time. And the way I do it is I say, this is going to be a major effect. And I wait for them to tell me, no, it's not. And then I know that this is going to be a good project. So I said, site size is going to be a big effect. We need to get smaller because I want to put more electrodes in. I want to have a person who can feel, I want someone who can see in megapixels, not kilopixels or pixels. I want many, many, many channels of information. And the only way to do that is to decrease the site size. So I said, unfortunately, when we start making the site sizes really small, I don't think they're going to work very well. We're going to start, I just felt like it's not going to work. And the student said, no, that's wrong. I said, all right, this is going to be fun. They were right. They usually are. So site size is not a major effect. We designed an electrode array that had 4 different site sizes. A very big one, 21,000, 7,200, 2,400, and 800. for benchmarking purposes, Nathan, who was getting stimulated when President Obama hit his glove, his electrodes are about 2,400. So we had electrodes that were much bigger and one electrode that was smaller. And it turned out, regardless of the site area, over time, all of the electrodes had very similar performances over time. And even if we teased it out for depth, we still saw the depth effect. Layer 1 got worse faster, but the electrode size in this range didn't matter. Okay, so that's cool. So that actually led us to this idea. Okay, let's change the design. See if we could start to work towards many, many small microelectrodes for eventual arrays of ultra microelectrodes. We know that design is important, and this is a very important paper from Matt Ward and Pedro Arathoki. I happen to be on it, but it was important without me. Looking at it was sort of the consumer reports of neural devices. Get every device they could get, including the Utah electrode array, Michigan electrode arrays. There's a Michigan electrode array. Every one that they could get their hands on and put them in. And the outcome is here. And I don't ask you to look at this graph very carefully, because really it just comes down to a quote from the paper. The results suggest significant variability within and between microelectrode types with no clear superior array. That sort of typifies our whole field for the past 25 years. Okay, so what can you do? Well, we've done a study where we took the electrodes and we moved them to the edge. If in fact, this is the design that we were set off on. Here's the substrate, and we put electrodes right down the middle, which is how they've always been designed. And we put them on the outside edge. We also put one at the tip, partly to compare the Utah electrode array effect to the Michigan electrode array effect. Nathan's electrodes are all down here at the tip. So this electrode allows us to compare that. Again, it's not a fair comparison because there's only a single tip electrode. There's 7 centers and there's 8 edges. If the reactive tissue response sets up like this, and we have a lot of reasons to think that it does, this suggests that the edge electrodes should be closer to healthy tissue than the center electrodes. I had the opportunity to present this at the University of Utah and tell Patrick Tresco he's right. This is his model. He told us this is how it's going to set up. He studied it for a long time. And it turns out that it's exactly right. The edge electrodes perform better than the center electrodes. Here's the edge. Here's the center as a function of time. And it was significantly different at every time point. We've also done this from a stimulation perspective. And in fact, the edge electrodes have lower thresholds at almost all depths. So it holds for stimulation as well. This has been, okay, so trying to get towards designing electrodes and thinking about reactive tissue response, could we make the electrodes really, really small? Other people started working on this in 2007. John Seymour and Daryl Kipke published this paper in 2007, where they had a microelectrode carrier with an ultra microelectrode outrigger. So this supported all the mechanical strength to get it in. And then they have these little extensions out here, which are 4 micron wide and 5 micron thick. In cross section, it looks like this, where you have the carrier and the ultra micro electrode array substrate. And it turns out the reactive tissue response surrounding the ultra substrate was almost non-existent. It gets small enough that it becomes stealth. This idea translated to a paper that TK Kozai published in 2012, a very important paper in our field, where they used carbon fiber electrodes that were less than 10 microns in diameter, strong enough to implant into the brain on their own, and they produced very robust recordings for a long duration. This has been picked up and carried for a long time by Cindy Chestick at the University of Michigan. She's been very successful in this program, making arrays of these. Here's a Tim Gardner's paper that does similar things, making these into high density arrays. This is Lan Luan's approach to doing this with microfabrication techniques. And then finally, you can even think to a certain extent, the NeuroPixels electrode array is sort of like this. You have high density arrays of ultra micro electrodes. In this case, the substrate is not ultra, although we're talking to them about redesigning that a bit as well. Okay, so if in fact we're going to go to many ultra micro electrodes, we know from basic physics that we're not going to get enough current out of a tiny electrode to get stimulation of neurons. We're going to have to use more than one electrode at a time. Could we use current steering techniques to stimulate 2 or 3 or 4 electrodes at a time to get a neuron to activate? And so the basic premise of that was this paper we published in 2002, where we stimulated the electrodes together in theory are doing something like this. The closer they are together, the more the fields overlap and collaborate to stimulate those neurons locally. As you pull them apart, at some point, they're working as 2 separate electrodes, not working synergistically at all. This is the basic data. Here's stimulation of a single electrode. Here's the 2 neighboring electrodes. And so basically you can see that 100 microns away, what happens is that mean is at 53%. So each electrode is being stimulated at 53% of what it was at a single electrode. What does that mean? That means that each electrode is stimulating with 47% lower peak, 47% less current. We have about 6% loss because it's 106% total current to stimulate to get threshold. So we're having a little bit of loss in that space, but we're able to lower each individual electrode amplitude. Okay, so all that put together is the premise for the current program that we're running, which is on high density ultra microstimulation. There's not a lot of outcomes here, and it's all unpublished, so I'm not going to delve into it too much, but I'll tell you a little bit about the program. It's a UO1 program. We're partnered with NeuroNexus first to look at running that site size study in a more reasonable level of scales. So in this case, we have large diameter electrodes at 50 microns. Those benchmark at 2,000 square microns. The biggest electrode we have is the same size that Nathan has in his brain. And then we go all the way down to 5 micron electrodes, which are 25 square microns. So that goes from something we know works in humans to something that's crazy. Like when we start designing this, people are like, what are you going to do with these 5 micron electrodes? We know we're going to have to change the surface chemistry to get the amount of current out of this. So we partnered with Stuart Kogan to put sputtered iridium oxide, sputtered ruthenium oxide, titanium nitride as an interesting control experiment to change electrochemistry. Interestingly, for those of you out there that are in this space, I'd love to talk to you about how all of a sudden capacitive electrodes are better when you're at the ultra scale. New stuff that's coming out all the time. And then mixed materials like polyethylene dioxide. So we're doing a lot of different material chemistry on those different sized electrodes, and it ends up being nonlinear with size. Some of the things that you typically think about at the micro scale, they switch when you get to the ultra scale. We're doing a lot of impedance spectroscopy with my MPI on the project, Mark Erasm. He wrote the textbook on electrochemistry or electrochemical impedance spectroscopy. I'm not using that lightly. There's a textbook in my office that he wrote. Warren Grill is on this project doing the modeling on the extracellular side. What we really want to do is have a theoretical model, which we could optimize, that would use impedance spectroscopy up to the edge of the electrode, switch to a voltage excursion in the extracellular space and cortical activation. And Warren's been working with his student to look at stimulation through volume to get a sense for what are we activating in what spaces. My lab is putting them all in and doing the electrophysiology, behavior, and histology. We have since moved on to design G2. We're rolling this out in 4 generations on our continual march to ultra scale electrodes on ultra scale substrates. We're taking the easy steps first, de-risking in pure DARPA sense. So we knew we could do G1. we had a pretty good sense we could do D2. In this case, we have arrays of ultra micro electrodes. And I don't have the data here, but I can tell you that we can easily stimulate 6 10-micron electrodes and get behavior. We're all the way down to, we can get 1 10-micron electrode excited activity, which translates to 5 5s. So if you stimulate 5 of these together at one time, we can get behavior. We haven't yet gotten 4. We're going to, but we haven't yet. All right. So let's spend a couple of minutes each on spine and nerve and bioelectronic medicine. I'm a central nervous system person. I always have been. Brain is where I grew up. But the nerve has a lot of powerful opportunities. And so, drugged by sponsors and the field, we've embraced the idea that we can take some of the things we've learned in the brain and develop new approaches in the nerve. In this case, we partnered with Jack Judy, Christine Schmidt, and Carlos Rinaldi to build a regenerative electrode. So these are microfabricated electrodes that are wrapped in a tissue engineered construct. We implant them into a nerve and see if the neurites will grow into the tissue engineered construct next to those microfabricated devices. We published this in 2002, showing the regeneration, and it actually produces really robust electrophysiology in this case. We've done quite a bit of spinal cord stimulation, which Renu called me out yesterday. This is all collaborative, but the idea that we know how to put electrodes in, we know how to measure performance as a function of time, we know how to apply energy safely. One of my collaborators is big in respiratory retrieval post spinal cord injury. So we've done a lot of spinal cord stimulation, epidural stimulation for closed loop for respiratory aspects. We've also had a long-standing program in situ development in this space. Can we build the electrode in the body? I'm not going to dwell on this too much, but you can use electrochemistry and these conductive polymers to build your electrode inside of the body. Okay, a brief foray into bioelectronic medicine. This is my motivation. And this is a graph. I'm not this crazy. I am a scientist, but I don't write down my weight every day. We have a Fitbit scale. It automatically logs it, right? This is my weight over the past 14 years of my life. And right here, that's 200, by the way, that's 210. Right here is when Jack Judy first talked to me about going to the University of Florida. Within 6 months, I had lost 60 pounds. My partner is a lab animal veterinarian. For those of you that do preclinical work, you know that the criteria for euthanasia is 20%. So after I recovered, she joked that I should have been euthanized. But it turns out insulin is important. And I was probably in the type 1 had started for me before this, but the stress of moving to the University of Florida really triggered the inflammatory cascade that took my pancreas down completely. And by the time I was diagnosed here, get insulin replacement therapy, get on shots, insulin's important. You can come right back up. It was really fortuitous in a weird way. And June 1st, 2014, Kevin Tracy published the editorial in a brand new journal, Bioelectronic Medicine. And he wrote on the revolutionary future of bioelectronic medicine. So the entire field of bioelectronic medicine was really just erupting, almost the same exact month that I was diagnosed. It also turned out that the lab moved right there 2 weeks later to the University of Florida. So I had startup, I had discretionary funds. Okay, let's go do something fun. Is the pancreas innervated? Really quickly, we answered that question. Yes, there are nerves that go to the pancreas. Are they motor or sensory nerves? There are motor nerves that go to the pancreas. You might ask why. Well, my argument in this space is that just like salivation in anticipation of eating. Your brain is preparing your digestive system for food. It's feed forward control. The brain programs the pancreas to release insulin in anticipation of food. So if we know that there's motor neurons going to the pancreas, can we modulate them? And can we result in insulin release in a type 1 model? Yes. We did selective neuromodulation of that nerve right there, going to the pancreas of a type 1 rat. We did continuous blood glucose measurement in those animals so we could watch their blood glucose. And I'm not going to go into the details, but basically you can see that we evoked insulin secretion in these type 1 animals. Very early data, just published last year. We're still working in this space, but there's a lot of promise. Okay, last bioelectronic medicine highlights. I'm almost done. I know it's super stressful to moderate. Pain modulation. We've done a lot of high frequency block of pain. This is not a new field, but we have contributed a little bit in this space. Hypertension. We published this paper just at the end of last year on stimulating the subdiaphragmatic vagus nerve to lower blood pressure in a chronically hypertensive rat. And learning, we ran a 4-year DARPA program stimulating the vagus nerve to improve learning and memory in a behaving rat model. This is the start of my acknowledgements. I'm very strong supporter of collaborative team-based science. I think what you're doing here is fantastic, phenomenal. I love that aspect of building diverse teams and coming at it from a very diverse population. This is our first DARPA team that I ran, and then the second one. And then this is the current U01 program with my MPI, our other PIs, and then NeuroNexus as a partner with all of the students that are on the team. So I'm very grateful to all of the collaborators that we've had. We've done a lot of different work across many different places in the nervous system I didn't think I was going to go. And it's really because of all of these people, my laboratory and collaborators that are that joined us for this picture, and there's some really phenomenal funding sources. Okay, last slide. I talked to Renu about doing this, and she approved it. So this is the first public announcement that I am moving. The lab is moving to Purdue. I started there. I'm going back. I was recruited back as the head of the department. I'll start there July 1st. I have opportunities for postdocs, graduate students, staff. I know that you're recruiting as well, and I'm not stealing, no stealing at all. But if you're a graduate student and you have a future in Indiana, or if you're a postdoc and you're transitioning out, please let me know. I'd be happy to talk about opportunities there. So thank you. $11:02.
So
10:02.
first, I think this is a big reveal. And so very big congratulations. Purdue is, as a college of engineering, it is very, very well recognized. And the Purdue department is also, the Biomedical Engineering department is also a very big department, over 30 faculty, I think, right? And they're linking you with the Indiana University School of Medicine. So you have a lot of stuff to go forward to do administratively.
I learned from watching the best.
But that's first a big congratulations on that. And then questions.